What we've done so far is we've looked at the process of gene expression, which involves inside the nucleus of a cell, we have the DNA. So this is a really simplified look here, but just to represent the DNA that we recognize as the chromosomes. The DNA has within it genes, so these are just sections of the DNA that code for RNA, which we said, and then can be expressed as proteins. The DNA is then copied into RNA in the nucleus, so DNA into single-stranded RNA, and then that RNA leaves the nucleus through nuclear pores. The RNA goes out into the cytoplasm, so we have our cytoplasm, and this is where the process of translation begins. Now, in translation, we have that little bit of RNA that could be the messenger RNA here. Then we have our ribosome that assembles along the messenger RNA, and then the polypeptide starts to be manufactured. Okay, so this is going to be our polypeptide, and that's our ribosome and RNA. Now, the first thing to bring up here now, as we continue along that process, we talked about the initiation, elongation, translation, the whole process itself. But now we're going to look at is what happens, kind of some other things that happen during that time and what can happen afterward. So this process is all taking place in the cytoplasm. Right? Now, the ribosome, we're assuming there's one messenger RNA, the one ribosome comes in, there's initiation, it reads to the end. But the thing is, this can happen simultaneously with multiple ribosomes. So we can actually have many ribosomes all working on the same polypeptide at the same time. Right? So one, say, if it was moving in this direction, this one would be further along, it'd be a bigger polypeptide. This one maybe is just getting started, it's a shorter polypeptide, but eventually they're all making the exact same polypeptide all at the same time in the cytoplasm, and we call that a polyribosome, showing that many copies of the same polypeptide or protein can be made simultaneously. Now, something else happens as well. When this process occurs, happening in the cytoplasm, it may begin and end, and that's it. And then when the process is over, one option is that the finished polypeptide is done. That's it. The finished polypeptide / now hopefully it becomes functional and we have a protein in the cytoplasm. Okay, so our protein is in the cytoplasm. But that's not always going to be the case. What's going to often happen is that the beginning of this, I'm going to kind of erase this and kind of redraw just a section of it. The beginning of this polypeptide, as it's being made, the first couple amino acids, so here's like an amino acid, an amino acid, an amino acid, an amino acid. This is all coming out of the ribosome, which is reading the messenger RNA. The first part, we're not going to get into the details here, that's something that could kind of beyond our class, what we need to cover. But the first few amino acids can form what's called a targeting signal. And in this case, we're going to talk about an ER targeting signal. So one way to think about this, and this is going to be the case with a lot of the first few amino acids, is they tend to be somewhat of an address. Now, the finished protein, it may work in the cytoplasm. Its job is as an enzyme, say, in the cytoplasm in metabolism. That could be the case. But you have to ask, what if the protein's job is to do something inside the ER itself, or inside the nucleus, where kind of this whole thing originated from, or in a lysosome, which is another compartment, organelle within the cell. What if it's a membrane protein? How do those membrane proteins that we covered earlier, the facilitated diffusion, transmembrane proteins, the proteins that were receptors involved in cell signaling. Those were transmembrane proteins. How do they get there? And what about the peripheral proteins too, the ones just attached to one side of the membrane? That has to happen at some point, so maybe it happens after this. The protein in the cytoplasm just kind of wanders around to where it's supposed to be. That's typically not the case. It has to be placed in the correct location. And for that, it has to have some sort of identity tag with it that says, this is where I belong. I belong as a transmembrane protein. I am part of the carrying out facilitated diffusion for the cell. So I need to get there. So for that to happen, there has to be some other mechanism of process. And you may have read this, and I've mentioned it before. We talked about other organelles within the cell. So the Golgi apparatus, which you've probably heard of several times and seen it written down, but maybe have no idea what it is or what it actually does. And the Endoplasmic Reticulum. So ER, since it's a long name, we often shorten it. So Endoplasmic Reticulum. And if you remember back to talking about the nuclear membrane, remember the nucleus of the cell has actually a double membrane, 2 phospholipid bilayers, an inner layer, and the outer layer. The outer layer is actually continuous with the ER. So here we actually have the ER, and these are representing Golgi, the Golgi apparatus. They're separate sacs, which we'll kind of get into in a second. So the first part of this is that some of the amino acids, the initial amino acids that have been assembled during the translation process, create a targeting signal. Out in the cytoplasm, we have, put this here, and I'm going to do this kind of crudely just to give you the basic idea. Something called SRP, which is a signal recognition particle. So SRP, the signal recognition particle. And what that's going to do is this particle here is a protein, and it's going to bind and identify this particular signal that says, I'm a protein who has to get inside the ER. So as protein synthesis is occurring out here in the cytoplasm, the signal is produced, and then this whole process, the ribosome, the messenger RNA, all of it is going to be moved to and attached to the ER membrane. The signal recognition particle is going to help guide this ribosome to the ER, and to here specifically a pore within the ER, which has a receptor for it. And so we're going to have this as our ribosome, like that. We'll get our messenger RNA, so I'm going to kind of draw it here upside down a little bit like this. So this is our messenger RNA. This is representing our ribosome. The transfer RNAs are they're in there. We can't really see them right now, but the transfer RNAs would be in here, binding, bringing in the new amino acids. And now what's going to happen is as the polypeptide is being built, it's going to be pushed through a pore. So this is representing a pore here in the membrane of the ER. So this here is representing the ER membrane, and this is called the ER lumen. It's the space inside the endoplasmic reticulum. So this is what we're talking about here. So this is the ER. The space here is the ER lumen. And what's happening is the ribosome, which is being attached to the surface of the ER, is then having the new polypeptide as it's being made push through a pore. And so this is our new polypeptide is entering the ER lumen. This is going to be necessary because this particular protein now has to be modified in some way, or it's going to be packaged up so that it can be transported to a unique location within the cell. All right, so the ER, one of its jobs, so one of the jobs of the ER is going to be to process new proteins. We'll talk about that in a second. And the other is going to be to package proteins for delivery. Right? So they're going to be delivered to the final location, and some of that's going to happen here in the ER as well. So take a step back. Protein synthesis, or the process of translation, happens in the cytoplasm. It may occur and just end there, and that's all there is to it. But in many cases, what happens is that the first few amino acids are going to code for a signal that has that particular ribosome with the growing polypeptide attached to it transported to the surface of the ER. So we have a type of the ER, sometimes we call this rough ER. And the reason we call this rough ER is because if you look at it under a microscope, the ER looks like it has studs on the surface of it. It has all these little circles attached to it. It looks rough. And if you wonder why does it look so rough like that? What are all those little bumps on the surface of the ER? And all these little bumps on the what we call rough ER are ribosomes. These are ribosomes attached to the surface of the ER, and so not just one. I'm doing one for the simplicity of the picture, but there would be many all over the surface. So there's many proteins being pushed into it, not necessarily the same protein either, a variety of different proteins that all require some sort of processing, not even the exact same, because the processing can be customized as well. So when this happens, eventually there's going to be a signal that stops it from being pushed into the ER, or just the protein ends and then it's going to be released. So that again is another option. So one option is the whole process occurs and then the new polypeptide, I'll just erase lumen here so we don't get that confused with what we have here. So this is the new polypeptide is now inside the ER lumen. All right. There's another thing that can happen here. And that is part of explaining how our transmembrane proteins get to be where they are. So what we have is, now we'll draw this here. This is one of these pores. Okay. The ribosome comes in. There is a messenger RNA. And a new polypeptide is built. Okay. And the polypeptide is being pushed through the pore. So this is the polypeptide into the ER lumen. Now, what can happen is, just like there was this signal here, a sequence of amino acids that said, say, take me to the ER, there can also be a sequence of amino acids, typically nonpolar amino acids, that are also something called a stop transfer signal. The stop transfer signal, so I'll write it down here, stop transfer signal. It's going to be yet another group of amino acids, and they're going to say, stop pushing me through this pore into the ER. And so what's going to happen is this process will continue, and some of it is here in the ER, but the rest of it now is outside the pore, and as it grows, it'll continue to grow, but not in the ER. It'll continue to grow kind of out here in the cytoplasm. So what do we end up with then? We end up with a protein that has part of it out on one side of a membrane. We have part of it that goes across the membrane, and then part of it on the other side. We have, right, you might recognize that as a transmembrane protein. All right, our transmembrane proteins, the pores, just like this pore here, that was a transmembrane protein that had to be assembled in this particular way. There are going to be sodium-potassium pump, receptors, all these transmembrane proteins. They will originate in a process like this, okay, where they get taken to the ER, they get inserted through a pore in the ER, and then at some point there's a stop transfer signal, which gets one section of that protein stuck in the ER membrane. And now it's just stuck here. And then we're going to get to later what's going to happen. So now we have a protein, let's say here, stuck in that membrane. This was a new protein. Maybe it stays there. That could be related to one of these pores in the ER. that binds signal recognition particles itself. But it maybe is some other kind of protein. Like I said, it may end up having to be out here in the outer cell membrane, and it's going to still have to get moved to that place. That's where the Golgi will come in. So protein synthesis begins. It moves to the ER. The proteins are pushed through the membrane of the ER into the lumen. Again, they may just get completely detached and then just be floating around in the lumen, or the process may stop. We also talked about multi-pass transmembrane protein. So if you remember the receptor that interacted with a G protein, that particular protein was a 7-pass transmembrane protein. So what that means is for it, there was a stop transfer signal that stopped the transfer, and then part of it was built then out here in the cytoplasm. There could then be later on down the line a start transfer signal again, and so now it's being pushed back into the ER. Okay? Ultimately, there could be multiple start and stop and start and stop transfer signals, and so this particular protein will be then woven through the membrane. So the stop transfers get them stuck, the start transfers get them pushed back through again, then eventually there's another stop transfer, and then we get some building in the cytoplasm, then it starts again, then it goes into the ER lumen, and so forth. So we get our transmembrane proteins of all different complexities built in this particular way. So that's the first part. We get the protein synthesis happening at the ER and or then continuing on, so the protein is pushed into the ER, and then we have our side option where proteins get stuck in the ER membrane and become the transmembrane proteins. Now what else happens in the ER? We said processing. Okay, so here in the ER are going to be a unique group of enzymes. So they're going to be enzymes in the ER. We're not going to go through all the specific details, but these proteins, now they have these signals attached to them. The role probably of that signal was just to get them to this location, just to tell them to be processed in a certain particular way. They're not going to be part of the final protein. So certain amino acids are cut off or removed. So when the polypeptide is made inside the ER, some of the enzymes will clip off amino acids and say, now we don't need that, we just needed it to get here, but now that we're here, we don't need it anymore. So they're removed. Some amino acids get modified. So this is another topic. We code for 20 different amino acids, right? We talked about that a little bit with the codons and the genetic code. But we could find in our cells over 100 different amino acids. So we say, well, where do they come from? Well, they typically all come from the core 20 that are modified chemically in some particular way. And again, we're not going to go into all the sorts of modifications, but some of the things that can happen are different functional groups could be added. Sugars could be added to them to form glycoproteins, right? And then there's going to be additional modifications that are going to be folding, right, which is what we can also have, which is going to be part of what occurs to this final protein. So remember, we had primary, which is just the sequence of amino acids. That's the peptide bonds. That happens with the ribosome. Their secondary structure, the alpha helices, and then the zigzag beta sheets, those typically occur spontaneously, hydrogen bonding between the different amino acids. But then we have three-dimensional shape, which could include ionic bonds and hydrogen bonds and a variety of other types of interactions. And those types of folding may need help. So here inside the ER, we often have another group of proteins called HSPs, or also referred to as chaperones. So in our case, we're going to refer to them more so as the chaperones. Now, HSP stands for heat shock protein, and this is sort of more how they were discovered. Certain proteins then denature when they are heated. Now, that means some of the bonds break, they change their shape, and then they lose their function. And they may then stay non-functional, and that's it for them. But some people found that some of these proteins that were deactivated through being denatured were able to then refold properly and then function once again. they found a correlation between the arrival of a new group of proteins that weren't there before the heat shock. So the heat, which shocked the cells and heated the proteins, before that happened, these HSPs weren't present in the cell. After that treatment, there were a lot of them. So the cells were triggered to produce sort of these stress proteins. When the cell's stressed, these molecules come and help protect the other proteins in the cell and can refold them back the way they should be. Now that's sort of an after the fact thing. What we're talking about is just the initial folding, the very first time it's folded. So you can think of these things as molecular chaperones, as sort of like an origami master who folds paper into some three-dimensional shape. They are going to grab on to these proteins, the chaperones, as they're being pushed into the ER, and they're going to help the three-dimensional folding occur in the proper way. So once again, there's a slightly more advanced topic of the different types of chaperones, the way in which they aid the folding process, and it's not something we're getting into. We're just introducing the topic. So within the ER, we can have a variety of types of chemical modifications. There is a three-dimensional folding of the polypeptides that's going to occur. Amino acids can be cut off here. So there's all these other things that are happening to get that polypeptide to its final form as a functional protein. The last thing we're going to get into with this is going to be the packaging part and the Golgi. We're going to keep that a little separate because I have to do a whole other drawing for it. So for now, just know the basic process of signal recognition particle, bringing the ribosome to the ER, that it gets inserted through the pore, that may just be the whole case, or it could be stopped and then form a transmembrane protein.